Hello students, in today's class we are going to learn about different sterilization and disinfection methods. So what is basically sterilization? It is defined as a process where all the living microorganisms including spores are killed. And for this we can make use of different types of methods such as physical, chemical and physiochemical. Disinfection is a process of eliminating most pathogenic microorganisms, excluding the bacterial spores which are present on inanimate objects. And here also we can make use of different types of methods such as chemical and physical. Here are some of the terms that are associated with respect to methods of sterilization and decontamination. So first term is decontamination. This is basically the process of removal of all the contaminating pathogenic microorganisms from articles by the process of sterilization or disinfection. Then is sanitization. This term is majorly used with reference to public health systems, and here it involves methods of chemical as well as mechanical cleansing. It is also associated usually with food industries, and the purpose here is to reduce the microbial load on the eating utensils to an acceptable and a safe level for public health. Then asepsis basically means means achieving an environment which is free of microbes, and it can be used by making use of various methods such as use of air filter, UV rays, etc. Antisepsis is basically use of antiseptics to uh, make our skin or the mucous membrane devoid of pathogenic microorganisms. The term bacteriostasis, so stasis here means static or stationary. So here we are making use of chemicals or whatever methods whereby multiplication of bacteria is inhibited, while bactericidal basically means killing, and here we are using various methods to actually kill the bacteria or microbes. So this chart talks about what are the various methods for chemical and physical sterilization and disinfection. They can be physical methods can be broadly categorized into heat-based methods, which are commonly used, which includes dry heat and moist heat sterilization. Dry heat sterilization is further categorized into red heat, flaming, incineration, use of hot air oven and infrared, while moist heat sterilization makes use of three different temperature ranges: below 100 degrees, at 100, and above 100 degrees for moist heat sterilization. Then radiations make use of either non-ionizing or ionizing radiation. And then filtration method is used for filtration of certain liquids or airs. Chemical sterilizing agents make use of chemicals which can be either liquid or gaseous in nature. So we'll be looking at all of these in more details. So the first category of methods is based on heat. So sunlight is the most common mode of sterilization, which is commonly used for like grains and stuff. And the mode of action is basically because of the excessive heat, it will cause protein denaturation and oxidative damage. Then other methods of dry heat sterilization includes red heat. So this is how we used to do uh, the red heat sterilization of the nichrome wire loop that is used for streaking purposes. Then flaming. So flaming basically means passing of the objects through the flame, and this is generally done for the mouth of test tubes and flask every time we open and close it. And incineration is basically destroying the articles at a very high temperature of about 1000 degrees Celsius using a specialized equipment which is known as an incinerator. And this is basically for completely destroying clothes or beddings in hospitals of persons who had been suffering from some kind of a highly infectious disease or a deadly disease. Then hot air oven, it is commonly used in the laboratories. So the different temperature ranges can be used. Commonly 60 minutes at 160 degrees Celsius is used. Or if you decrease the temperature, then the increase the temperature, then the time required for sterilization will comparatively decrease, as you can see. Advantages: it is effective method of sterilization of heat stable articles such as glasswares, or sterilizing of oils and powders can be done by making use of hot air oven. Disadvantage is cotton wool or paper may slightly get charred, and sometimes you will have a smoky smell on the glasswares. And it one of the disadvantages also, it takes longer time compared to autoclaving. Then we have infrared rays, and infrared rays bring about sterilization again by generation of heat. And so basically you have a conveyor belt that is moving, and whatever articles you want to sterilize using infrared, they are basically passed through with the help of a conveyor belt. They are passed through a tunnel that is heated by infrared radiators to a very high temperature of about 180 degrees Celsius. So articles that are commonly sterilized includes metallic instruments and glasswares. So one of the challenge with this is that it requires specialized equipment that will generate the heat because of infrared radiations. Then moist heat sterilization. So this is based on use of water in sterilization. So three temperature ranges are commonly used. You can use it below 100 degrees Celsius. The process is known as pasteurization. This is commonly used in food and dairy industry. In case of, for example, milk, two different methods for pasteurization are commonly used, like or temperature range. Either you are heating your food products at 63 degrees Celsius for 30 minutes, or the other method is known as a flash method, where you heat at a slightly higher temperature, that is 72 degrees Celsius, for 15 seconds. And then followed by this, they are quickly cooled. So this method is suitable for destroying most milk-borne pathogens. However, it is not a complete disinfection method. So most pathogenic organisms will be killed here. Then we have vaccine baths. So here contaminating bacteria in vaccine preparations can be inactivated by heating in a water bath at 60 degrees Celsius for an hour. So here only vegetative cells are killed, while the spores will survive because they can tolerate higher temperatures, even above 100 degrees. Then we have serum bath. So this is for decontamination of serum, which is used. So water bath at 56 degrees Celsius for 1 hour is commonly used for several successive days. So here we are using low temperature again, not too high temperature, because we don't want to inactivate the proteins. So again, only the vegetative cells are killed while the spores will survive. The second method is at a temperature of 100 degrees Celsius. So commonly for household purposes, even boiling of water is done for sterilization. So you can also do it in an autoclave, which is also known as steaming at 100 degrees Celsius. So this you can do if you keep the pressure knob on. So in that case, the temperature of autoclave is only around 100 degrees Celsius, and also is known as steam sterilization. It is generally used for those media that cannot be autoclaved at very high temperature, especially media containing sugar and gelatin. And sometimes you can also use a method of steaming for 20 minutes for 3 successive days. Like the first day you do the steaming, and then you incubate the media for 1 day, and then second day you do the steaming again, and then you just wait for 1 day, and then third day you do the steaming again. So here basically what happens, if there are any spores, then because of the exposure to high temperature, that is 100 degrees Celsius, so that comes like a stimulation. So after the normal day incubation or normal temperature incubation, at that time the spores will germinate. And then when we do the steaming again for second days, they are in the form of a vegetative cells, so they will be killed. So this method is also known as sterilization. Then thirdly, we make use of a temperature above 100 degrees Celsius, that is used in an autoclave, where we make use of 15 lbs pressure for at least 15 minutes at 121 degrees Celsius. This is a diagram of a typical autoclave. Then we can also make use of non-ionizing radiations. So UV rays are commonly used for sterilization, and the most effective UV rays are about 260 nanometer. They are generated by making use of high pressure mercury vapor lamp. And UV radiations will basically induce formation of thymine dimers, and as a result of which the DNA replication process is inhibited. So these can be effective against bacteria, viruses, yeast, basically vegetative cells, but they are not going to kill the spores. Then we have ionizing radiations. So electromagnetic rays such as gamma rays can be used for ionizing radiations. So radioactive isotopes such as cobalt-60 or cesium-137 can be used. They have more penetration power, so less exposure time is required in case of ionizing radiations. These are capable of killing bacteria, fungi, viruses, and even spores. They are used in commercially for sterilization of disposable petri dishes, syringes, antibiotics, hormones, etc. One of the disadvantages is that they cannot be actually switched off like a machine. It is basically making use of radioactivity. So whenever the activity is gone, only after that the effect is gone. One of the disadvantages is that the glassware, they generally tend to become brownish because of exposure to the gamma radiations, and also the fabric can lose its tensile strength, and sometimes the food also can have some not very good flavor because of gamma radiations. Then filtration is another method. In the laboratories, we have already looked at laminar airflow, which makes use of HEPA filters for filtration of microbes, generating a sterile environment where you can work. Laminar airflows also have presence of UV light that will also help in uh, uh, killing the vegetative cells. Then this apparatus that you can see is of a membrane filtration unit. It consists of like this is also similar structure. It consists of a lower flask or a container and an upper container in which you put your sample that you want to sterilized. And in between the two is present a membrane which has a pore size of about 0.015 micrometer to 12 micrometers, so which is very small compared to even the size of a bacterial cell. And then this is connected to a vacuum filter through the pipe. So the when we this is connected to a pump basically. So when the pump is switched on, it is going to create suction. As a result, whatever liquid is present on the top, it will be filtered through the membrane that is present in between and will be collected in the lower container. So you can store it in the same container or in any other sterile container and use it as and when required. So this is generally for those medias which cannot be autoclaved. For example, generally the animal cell culture medias, these need to be sterilized in this method. Then these type of filters that you see are known as syringe filters because they are used in combination with needle with the syringe basically. And you attach the syringe filters to the mouth of the syringe, and this can be commonly used for filtering out small volumes of liquids such as antibiotic solution, which also cannot be sterilized by any other method. Then we have chemical sterilizing agents. So alcohol is the most common chemical sterilizing agent that is used. It will basically dehydrate the cells, disrupt the membranes, and coagulate the proteins. Examples include ethyl alcohol, methyl alcohol, etc. 70% alcohol solution is commonly used as an antiseptic on skin. It can be used for surface sterilization. Disadvantage includes it is a skin irritant and it is volatile and inflammable. Then aldehydes are other kinds of agents that can be commonly used for chemical sterilization. They act through alkylation of amino carboxylic or hydroxyl groups, damaging the nucleic acid. Examples include formaldehyde, glutaraldehyde. 40% formaldehyde solution is commonly used for disinfection of operation theaters, biosafety cabinets, fumigation of labs, etc. So, exposure for at least 3 hours at alkaline pH is required for the action of glutaraldehyde. 2% formaldehyde solution at 40 degrees Celsius for 20 minutes is used to disinfect wool, and 0.25% at 60 degrees Celsius for 6 hours to disinfect animal hair and bristles. Disadvantages, the vapors are irritating and can be even irritating to your eyes, and they have low penetration power. It also leaves a non-volatile residue on the surfaces which are sterilized. Then phenol, it is basically inactivating the proteins. Okay, and examples include 5% phenol, cresol, lysol solutions. They are generally bactericidal, fungicidal, or mycobactericidal in nature. However, they are generally not functional against spores and viruses. They are highly corrosive in nature and they are generally used for disinfection of hospital floors, laboratories, etc. Disadvantages, they are toxic and corrosive in nature. Halogens are also commonly used as sterilizing agents. They are oxidizing agents and cause damage by oxidation of certain sulfhydryl groups of enzymes. Chlorine reacts with water to form hypochlorous acid, which is microbiocidal. They are also commonly used in like swimming pools and all. They are commonly used. Tincture of iodine, generally 2% iodine in 70% alcohol is used as an antiseptic. Chlorine gas is used to bleach water. Then one of the disadvantages of these molecules, they are rapidly inactivated in presence of organic matter. Iodine is also corrosive and can also stain different articles. Then heavy metals, they act by precipitation of proteins and oxidation of sulfhydryl groups. They're bacteriostatic in nature. Different salts of mercury or mercury chloride, silver nitrate, copper sulfate, they are commonly used heavy metals. Silver sulfadiazine is used topically to prevent colonization and infection of burn tissues. Mercury salts are active against viruses at a dilution of about 1 to 500 to 1 to 1000. Copper salts can be used as a fungicide. Disadvantages includes that mercury chloride is highly toxic and are readily inactivated by organic matter. Hydrogen peroxide is another chemical sterilizing agent that functions by release of nascent oxygen. This oxygen can damage proteins and DNA. It is generally used at 6% concentration to decontaminate instruments, various equipment such as even your biosafety cabinets can be, the walls of biosafety cabinets can be wiped with hydrogen peroxide. Strong solutions are sporicidal, which is advantageous. One of the disadvantages is that it gets easily decomposed by light and by activity of enzyme catalase. And proteinaceous organic matter will reduce its activity. Ethylene oxide is an alkylating agent. It functions by alkylating sulfhydryl amino carboxyl and hydroxyl groups. It is a cyclic molecule which is colorless liquid at room temperature. It has a small ether-like order that readily polymerizes and is flammable. Uh, since it is flammable, it is generally used not directly, but in a form of a mixture with carbon dioxide. It is used to sterilize heat labile articles such as bedding, textiles, rubber, plastic syringes, petri plates, etc. One of the disadvantages, it is toxic, irritating to eyes, and highly inflammable. It is also mutagenic and carcinogenic. So that's it about various physical and chemical sterilization methods. Kindly like, share, and subscribe if you like the contents and to get immediate updates.